Mrs. Vance again, and this is Unit 5, Part 2 notes about homeostasis and membrane transport. So we talked about the cell membrane and its structure in the last part of notes. Now we're going to talk about how the membrane helps maintain homeostasis. So remember from middle school science that homeostasis means that living things have to maintain a stable internal environment. And there are various ways to do this. Sometimes it has to do with behavior and various things like that, but most of the time we can depend on our membranes to take care of most of that for us. The thing that you need to realize is that most activities in organisms, and therefore in their cells, are directed toward maintaining homeostasis, okay? And because the membrane is in control of what enters and leaves the cell, it does a very important part of that job in helping cells stay in a stable, steady state all the time. And so we're talking about moving things into and out of the cell and controlling that, which is what the membrane does. So how do cells do that? Well, there are several different processes that are involved in this, and they can be divided into two major categories, and those are passive transport and active transport. Passive transport doesn't require any energy. These are things that will happen on their own without any input of the energy by the cell. These are things like diffusion, osmosis, and something called facilitated diffusion that we'll talk about in a few minutes. The second major category is active transport. These are methods of movement of things into and out of the cells that require energy, that require the cell to use some energy to do those jobs. Some of the proteins that are embedded in the membrane use energy to move molecules either against their concentration gradient or in a large capacity like endocytosis and exocytosis that are involved in moving larger particles into and out of the cell. So we'll talk about each one of those in turn. Diffusion is the main form of passive transport. Okay, diffusion by definition is the movement of molecules from an area of high concentration to an area of lower concentration. And this is what's referred to as moving down the molecule's concentration gradient. And so if you look at the diagram up here in the upper right, you'll see that we have a high concentration of molecules right here and less or none over here in this part. And this is kind of like when you drop a droplet of food coloring into a glass or a container of water. You can, at first when you do that, you can see the droplet kind of stay together. And over time, what you start to see is the molecules of the dye start to spread out from that original drop and go into the part of the water that wasn't colored before. And after a few minutes, what you'll see is the water all maintains the same color because those molecules have spread out evenly in the area there. So we can look at it like this. At the bottom, we have down here the same thing that we saw in the top, except now we have a membrane here. And so membranes are not totally impervious to anything moving through. They generally have pores of some kind. Sometimes they're bigger, sometimes they're smaller. But if you have those molecules, same molecules of dye there in a container that has a membrane in the middle, what you'll see is, first of all of the molecules of dye will stay on one side, but over time, as those molecules bounce around, as molecules have a tendency to do, they don't sit still, then some of them are going to pass through the membrane, and over time, some will pass through from this side to the other side, or back the other direction, but eventually you'll get them spread out into a pretty evenly distributed mess of molecules that we would call equilibrium, that we would say it reached equilibrium at that point, and both sides of the membrane would have the same amount of molecules, and so therefore the dye would be evenly dispersed within the container. Let's watch a quick video showing how this works.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So there are some molecules that can easily move this way, and others need a little bit of help along the way. Okay? Osmosis is a type of diffusion that specifically talks about the diffusion of water. And so this is the diffusion of water down its concentration gradient from a high concentration of water to a lower concentration. And as it diffuses across that membrane the membrane that is selectively permeable. And so here, it's passive transport, it's movement of molecules from an area of high concentration to lower. It's just specifically talking about water molecules moving across a membrane. This is really important because cells, the cytoplasm within a cell is a watery substance, and cells generally are in a watery environment. And so if there's more water or less water in the environment, then that could affect what happens to the cell in response to that, because the water molecules are going to want, they're not going to say, oops, sorry, I don't need to go in there. They're going to move on their own, and this is going to happen naturally without any expenditure of energy. So when we talk about water balance, we also talk about the concentration of solutes. Solutes are the things that are dissolved in water, okay? And so we use a term called tonicity to talk about the ability of the solution to cause the cell to gain or lose water. A hypotonic solution is one that causes a cell to take in water. It has a lower concentration of solutes than the cell has. That hypo part of the word means under or lower. And so if you have less solute dissolved in the solution around the cell than you have within the cell, that's going to cause water to move into the cell, and it's going to cause the cell to swell, sort of like if you overfill a water balloon, okay? An isotonic solution, iso means the same, and this is a solution that doesn't cause any net movement of water into or out of the cell. The water molecules are always going to be moving. Some will move in, some will move out, but you won't have a gain if water molecules in one place or the other. Basically, an isotonic solution has the same concentration of solutes that the cell has, and therefore the cell is going to remain the same size, and it's not going to swell, and it's not going to shrink. The third kind of solution is called a hypertonic solution, and hyper, again, means too much or more. And so this is a solution that's going to have a higher concentration of solutes than the cell, and therefore it's going to cause the cell to lose water. And if the cell loses water, it's going to shrink in size, kind of like if you took that same water balloon we talked about before and stick a pin in it and let some of the water come out, it's going to shrink. When we look at cells in hypertonic, isotonic, and hypotonic solutions, we can see what happens here in different kinds of cells. In the top row, we're looking at animal cells. In this case, it's looking at red blood cells. And so if you place the animal cells in a hypotonic solution, meaning that there is less solute dissolved in the surrounding solution than there is dissolved in the cell, that's going to cause water to move into the cell, which is going to cause the cell to enlarge or swell, and eventually it's going to reach the point where the membrane can't hold that volume anymore, and it's going to rupture or lyse. Lyse, remember, means break down. In an isotonic solution, the animal cell is going to have water moving equally into and out of the cell, so it's going to maintain its normal shape and size. Whereas in a hypertonic solution with a high solute level, it's going to cause water to move out of the cell into the solution, and that's going to cause the cell to shrivel or shrink up. In plant cells, it works a little bit different because plant cells have that rigid cell wall that kind of maintain their structure. They also have that central vacuole that can take up some of the excess water. Plant cells actually like to be in hypotonic solutions because that's what helps them maintain their structure. When you see plants standing up straight and tall, that's because they've had plenty of water and their cells are in what we call a turgid state, in a hypotonic state. They've got plenty of water in there. There's pressure of the water against the cell wall, and that maintains that nice straight posture that we expect our plants to have. If you haven't watered the plant in a while, sometimes it starts getting kind of soft and starts wilting a little bit, and that would be what happens in an isotonic solution. You end up with cells being what we call flaccid. They're not quite as stiff and structured as they would be. They're not in that turgid state that they would be in a hypotonic solution. And so the plant will start wilting a little bit if it's in an isotonic solution. And finally, if you put a plant cell into a hypertonic solution with a lot of solute outside the membrane, that's going to cause the cell to lose water, and that's going to cause shriveling or what we call the cell being plasmolized as the cell membrane actually starts pulling away from the cell wall, and you can see definitely that the cell contents have been shrunk compared to the normal size and shape of the cell. Now, there's another kind of movement of water into and out of, or other molecules into and out of cells. This is called facilitated diffusion. This is also a passive process, but sometimes you have molecules that are too large, or perhaps they have a positive or negative charge and can't pass through the phospholipid bilayer. Remember, in the phospholipid bilayer, you've got your polar heads that are not going to react well with nonpolar substances, and your nonpolar tails that are not going to like polar substances. And so if there's something that has a lot of polarity, like let's say water, okay, or other kinds of molecules like that that need to come in and out of the cell, you need some kind of a channel for them to pass through. They still are diffusing. They're moving from an area of higher concentration to an area of lower concentration, but they're moving through a channel, and that's because it's facilitated or made easier for it to happen. If you think to your most of you have taken Spanish or learned some Spanish, and the first part of this word, F-A-C-I-L, is actually in Spanish is the word fácil, which means easy. And so facilitated diffusion is diffusion that is helped or made easier by the presence of that protein. There are lots of different things that come into the cell or leave the cell via facilitated diffusion. Water is one of those things. Water molecules are pretty small, and they can move through the membrane somewhat, but they can move faster in and out of the cell via special channels called aquaporins that allow the water to move. And again, this is also a passive transport because no energy is needed for this to happen. But what if you have something that you need to move out, regardless of how much there is into or out of the cell? If you need to move it out or in against that concentration gradient, then you need to do something called active transport. In active transport, molecules are moved against their concentration gradient, and this requires energy. It's kind of like passive transport is kind of like coasting your bicycle down a hill. Active transport is like pumping the bicycle up the hill. In other words, it takes more energy to get up the hill than it is to get down the hill. And so we think about they're moving against their concentration gradient, like you're moving against the gradient of the hill when you're riding your bike up the hill. That's going to take more energy than it does going down the concentration gradient. So these molecules are moving from an area of higher concentration, from an area of lower concentration to an area of higher concentration, because we need those particular molecules in the cell. This does require energy, and the energy is in a molecule called ATP that we're going to learn a little bit more about coming up really soon. There are some of these proteins that are called pumps. They're transport proteins that actively transport small molecules and ions that require energy to do the work. An example of this is something called the sodium-potassium pump. The sodium-potassium pump is needed to maintain the concentration of sodium and potassium ions across membranes like muscle cells and nerve cells that have to maintain a difference in charge between one side and the other. And so you have the sodium-potassium pump that pumps 3 sodiums out of the cell. It opens up this way so sodium ions can go in there. For the movement to happen, you have to transfer a phosphate off of ATP to the molecule. When you do that, that causes the molecule to change shape, like you see here, and it releases the sodium ions, and then there's a place that potassium ions can bind. And when the potassium ions bind, that causes the cell to change its shape again and bring the potassium ions in. It requires energy in the form of ATP. Now, what if you have things that are really big that you need to move into or out of the cell? These are large molecules or multiple copies of a molecule. These are other types of active transport that are involved in bulk transport. The first one is endocytosis. This brings large materials into the cell. And basically what happens is the cell membrane kind of pinches in to form a vesicle enclosing the particles. Now, there are 2 types of endocytosis that we discuss all the time. One is called phagocytosis or phagocytosis, and this is called cellular eating. The phago part means eating, okay? And this is bringing in larger particles into the cell. This is what amoebas do when they eat bacteria or other small cells within their environment. This is what your white blood cells do when they're eating the bacteria or other molecules that can be harmful to your body. The second kind of endocytosis is penocytosis or pinocytosis, and this is cellular drinking. The peno part refers to drinking or bringing in liquid, and this brings in smaller particles or droplets of liquid into the cell. The other kind of bulk transport is exocytosis. This is when you have larger molecules that need to be sent out of the cell. if the cell is one that makes something like hormones or other kinds of chemicals that need to be released into the bloodstream, for instance, they will be made in the ER and processed in the Golgi apparatus into vesicles. Those vesicles will be stored till they're needed, and then the vesicle can fuse with the cell membrane to expel the contents out of the cell. It's basically the opposite of endocytosis. So the white blood cells, of course, would use endocytosis or phagocytosis to remove bacterial cells from the body, and then any kind of large waste particles or products of the metabolism of the cell could be released by exocytosis. I will post some other videos into Schoology for you so that you can see some examples of how these things work.